researching early Reccembran fauna from the Moshucene, for a long time we were puzzled by the sudden appearance of a seemingly totally new clade, unlike anything seen previously. They possessed a central area we call a head, surrounded by two appendages which could vary drastically in appearance, from seemingly countless tentacle-like bristles to long and slender tubes which made these creatures look more like artificial creations than exoanimals. And that's without even talking about the weirdest of them all. It took us quite a lot of time to understand the fact that we were looking at a case of extreme neoteny. Some instances of neoteny had been observed previously in Reccembran history, organisms which didn't fully develop, but kept being able to reproduce. But this was brand new. We thought this level of developmental impediment would surely kill any organism, but somehow it did not. Exoanimals develop in 3 stages. First, a tiny central tube forms, sometimes made from a single row of compartments. Then it grows to stems, which start creating the outer tube-like structures. In tuboids, that's as far as it gets, but in exoanimals like eelworms, this outer shell becomes the skin, which then grows inwards. The tubes create more cells as well during this stage, to fill the organism with the required tissues. It was this last process which was interrupted in the case of the Halflings, as we've come to call them. In Halflings, the central tube is much larger, made up of many more compartments than in all other exoanimals, and the 2 stems grow into 2 distinct halves of the tube skin, which never meet. These half cylinders then fill up with tissues and are enclosed by the layer of skin growing above them, which loops back into itself as if creating 2 separate tuboids attached to the central tube, or the head of the halfling. Halflings were mostly planktonic. In fact, at first we didn't even realize we were finding fossils of them until we started looking at the minute details next to larger fossilized organisms. From what we have managed to observe, there were 3 groups we can easily classify them into based on their appearance, but their taxonomical relatedness is unclear, and we could be classifying unrelated species as belonging to the same clade. Based on their appearance alone, the first group of halflings are the Papusiforms. Their name comes from the resemblance of their lateral tubes to the papus found in many plant seeds. These halflings were likely filter feeders, catching even smaller food items such as ball grasses and developing organisms, and digesting them by secreting enzymes out of the setae the lateral tubes were made of. About the reproduction, we just have to assume they reproduce via broadcast spawning like most exoanimals at the time, although no clear evidence of a reproductive patch like that of the eelworms has been found. The second clade was comprised of the H-fins, named so because of their shape. They are assumed to be the most basal halflings, with the lateral tubes being just that. 2 tubes, mostly filled with muscle fibers and potentially 2 separate digestive glands connected to their surfaces, which they probably use to absorb digested material. They also seem to have many setae on their tubes, and judging from the amount of organic debris collected around them in fossils, they were likely quite sticky. The third clade is by far the weirdest, and they might as well have been called the Quadlings, for they are only 1/2 of a halfling. Imposter jellyfish, or Gellims as they called them, have a really strange and interesting life cycle. They start out similarly to Papusiforms, but with the lateral tubes divided into clearer broad segments rather than filaments. They had 11 of these segments on each lateral tube. This larva, weirdly enough, did not use its lateral tubes for eating, but for locomotion, opening and closing their segments like an umbrella around their head. It was this head which was used for eating, by producing digestive enzymes from its skin and absorbing the liquefied matter. But this isn't the interesting thing about Gellims, not by far. Once a larva grew large enough, which could be very large compared to, say, a Papusiform, it would split in 2. How they did this is still unknown, but we suppose the middle of the head simply started growing thinner and thinner until the 2 halves snapped off. The 2 new individuals, both identical clones of each other, would then go on to grow even larger and reproduce, creating more larvae. And that's all on early halflings for now. This is Hnrrienkast signing off.